So it's sometimes called aerobic respiration because it takes place in the presence of oxygen. So oxygen means it's aerobic. And the whole point of this process is to produce ATP from the foods that we ingest. And this is really common. Happens in animals and most plants. And in addition to having ATP, we're also going to be making some other substances, such as water and carbon dioxide as waste products. So if we look at the overall equation for the process of cellular respiration, the basics of it, we start with glucose and we start with oxygen. Okay? And then at the end of this whole multi-step process involving multiple locations in the cell, we're going to end up with carbon dioxide and water as waste products and the ATP that we need. And so this is going to have three very distinct steps, as well as a little linkage between the first two steps. Okay. And so here's just kind of like an overview of what it is, showing that the first step takes place in the cytoplasm. And then if there's oxygen, we will move into the mitochondria for the linking step and the last two steps. And typically in a perfect world, we would end up with 36 ATP. So what is ATP? Well, that's the abbreviation for adenosine triphosphate. And adenosine is basically the adenine that you learned about when you learned about RNA and DNA, and it's attached to a ribose. And then to that, we can have up to 3 in organic phosphates. So it's the PO4, no carbons attached to it. So there's a little bit of energy when the first phosphate attaches that gets stored in that bond. There's a lot more energy in the second bond, and that third phosphate stores a ton of energy by comparison. So we're making the ATP because we want to use the ATP, which means we want to break it down back to the ADP and release this humongous amount of energy that was stored. So stop and think for a second from what we've already learned. What would we have to add to ATP in order to break this down? And is this reaction an example of dehydration synthesis or hydrolysis? So if you can figure out which reaction it is, then you can figure out what needs to be added. Okay. Yes, so it's water that needs to be added because this is hydrolysis, a hydrolysis, where we are breaking the bond, we are dephosphorylating the ATP to release the inorganic phosphate because we want the energy that's stored in that bond. So that's the lysis part. The hydro part is adding the water. So let's get back to cellular respiration and the 3 main steps. The first one is glycolysis. So what does that mean? That means we're going to start with the glucose molecule. This is going to happen in the cytoplasm. And then if there is oxygen, the end products of glycolysis will then move across the membranes of the mitochondria into the mitochondrial matrix, where the intermediate stage is going to occur, as well as the second step, which is the citric acid cycle, or you may have learned it as the Kreb cycle or the TCA cycle. They are all synonymous with each other. But that part takes place in the mitochondrial matrix after that intermediate step. Here's the little intermediate step right there. And then we are going to stay in the mitochondria, but move to a third location, and that is the cristae, these guys, those little shells, those little ledges, for our third step, which is the electron transfer chain, at the end of which we have oxidative phosphorylation. And that actually uses the space in here as well as the edge. So we'll see that in more detail later on. So let's get started with step number 1, which is glycolysis. Glyco for glucose, sugar, and breaking. So no oxygen is required for this part. This part occurs in the cytoplasm. There's 10 steps. You don't have to know them. Basically, we're going to start with one glucose, which is a 6-carbon sugar. Remember, C6H12O6. And we're going to break it down into 2 pieces. So we're going to take the 6 carbons and put 3 here and 3 in this molecule. And the molecules that we're going to end up with are called pyruvate or pyruvic acid. Now, in order to break down a bond, that's going to require some energy. So we are actually going to be using some ATP at the beginning. And once we have our 3 carbon pyruvic acids at the end, we also will have made a few other things that exit this process. We're going to make 4 ATPs, and we're also going to make 2 compounds known as NADH₂. Okay, there should be a 2 on the end. No, there shouldn't, just NADH. The 2 here is an error. Oh, I should have fixed that on the picture. Okay, and so the net result at the end of this is sugar and energy go in. Pyruvic acid, 2 of them come out, a net of 2 ATP because 2 had to go in and 4 were made, so you end up with 2. And then the 2 NADH. And all I want to say about NADH right now, this is a molecule that's an electron carrier. Okay. So in the pathway, we start with the energy investment, putting in the ATP. And then we go to the parts that are giving us our compounds, some which have energy, some of which are ATP, and the other ones are NADPH over there. So we're going to have our 2 high energy compounds. Here are all the steps and the shapes of the different molecules. We don't have to learn that in this class. But I just want to show you, here's an ATP going in, here's an ATP coming out, here's an NADH coming out, here is ATP going out, and here's ATP going out. So do you notice this first part up here is in blue because up here, everything is still a 6-carbon sugar, and then we break it down into a 3 carbons. So once we have the 3 carbons, you have to realize the rest of glycolysis, each of those 3 carbons is doing the exact same thing. So each one of them is doing this bottom part transition. And that's why at each step, we end up having 2 NADHs, 2 ATPs, and 2 ATPs, because each one is generating 1. What I do want to point out way up here in this early step, the second primary reaction takes that second ATP, the enzyme phosphofructokinase 1 is the enzyme that's required for this second chemical reaction to occur. And this is the site where glycolysis is regulated the most through the process of cellular feedback. Here's that same enzyme right here, phosphofructokinase. If we have a lot of cellular ATP, that ATP binding will inhibit kinase activity. So this phosphofructokinase is a kinase. Okay, so we are going to be inhibited. So if the cell has a lot of ATP, then this enzyme is not going to work. On the other hand, if we have low cellular ATP, then we definitely are going to increase our glycolysis. So that basically is where the cell decides, do I want to do this now, or are we going on a little siesta break? There is an endocrine regulation of this as well, because we are starting with glucose, and insulin and glucagon regulate blood glucose levels. So depending on the cellular levels of glucose and fructose, we will either increase or decrease it. But we will come back in the endocrine system to talk about glucose and fructose. You don't have to worry about that at all. You also don't have to know phosphofructokinase, the name of that, but just realize it's near the beginning that we can stop it if we already have too much ATP in the cell. So once we have our 2 pyruvic acids at the end, now we are at a fork in the road. Do we go right or do we go left? Well, if we have oxygen, that's going to make a difference. We're going to go to the right side and we are going to have an aerobic thing happen. So the pyruvic acid, the products of glycolysis will move inside the mitochondria, and then we can continue on with our linking reaction and the Krebs cycle and the electron transfer chain. If we don't have oxygen, then we're going to take the left branch, and the left branch, the only thing we can do is fermentation. We'll come back near the end and talk about fermentation very briefly. But let's assume we have oxygen and we're going to continue on to the linking reaction. Sorry. So here we are in the cytosol with our pyruvate, and what happens is in this linking reaction, we are going to have a redox reaction where we're going to have a reduction as well as an oxidation. And this occurs in the mitochondrial matrix, which is kind of like the cytosol of the mitochondria, but because mitochondria is not the cell, we don't call it a cytosol, we call it the matrix. And so basically, we're going to do 3 different things. The first thing, we start with our 3-carbon pyruvate, and we are going to take off our carboxyl group. A carboxyl group is just CO2, so it's going to be carbon dioxide. It's going to be released when we reduce NAD and form an NADH. So we're going to form another electron carrier in the linking reaction. No ATP directly, but an electron carrier. And the third thing that happens is there's a coenzyme called coenzyme A that's required for this reaction to occur. And what coenzyme A ends up doing, it actually binds to this 2-carbon piece over here, which is an acetyl group. And so we end up with something we call acetyl-CoA. So basically in the linking reaction, we start with pyruvate. We make an high energy carrier, we release some CO2, and we end up with acetyl-CoA. Now recall, since we end up with 2 pyruvates for 1 glucose molecule, this is going to happen twice, so 2 NADHs would be produced. All right. Now we're into the second step, the citric acid cycle or the Krebs cycle, where we're going to make more ATP, more electron carriers. This is still in the matrix. It's got a bunch of enzymes. And if we look at this whole thing, what you're going to see is because we are going to start with some small 2-carbon things, and we're actually going to be making longer carbon molecules, we are going to have to add some water, and we are going to make 2 different types of electron carriers. We're going to make some more NADH, but we're also going to make some FADH₂, as well as making some ATP directly. So this just kind of shows you, here was our pyruvate at the end of glycolysis, here was at the end of our linking reaction where we had acetyl-CoA. The very first step, we're dropping off the CoA, and we are adding these 2 to the 4 at the end of the citric acid cycle. So now we have citric acid being formed. Then we make electron carriers, more electron carriers, some ATP directly, more electron carriers, and more electron carriers. And in doing that, because we're blowing off a couple of CO2s, we end up with a 4-carbon molecule at the end there. So realize once again, we have 2 citric acid cycles for every glucose molecule. So at the end of 2, we've released 4 carbon dioxides. We've made 2 ATP directives. We've made 2 of the FADH₂ electron carriers and 6 of the NADH electron carriers. So how is this regulated? It's primarily at this very first step where we are going to make the 6-carbon citrate. We've got that 2-carbon thing going in. Once again, if there already is a lot of NADH and ATP in the cell, then demand for more energy is very low, and we will not have very much of this pathway occur. However, if we have low levels of ATP and low levels of NADH and FADH₂, then we have lots of demand for make more energy. And so the enzyme that's synthesizing citrate from the 2-carbon acetyl-CoA and the 4-carbon ending product of the cycle, that increases its activity. So by now you must be asking, what on earth are NADH and FADH₂? Well, they have names. You don't have to know what they are, but realize that NADH was made in both glycolysis and the Kreb cycle, but the FADH₂ was only made in the Kreb cycle. And as I said, both of these molecules like to carry a bunch of electrons. Basically, they're going to be acting like a coenzyme. And in the third process of cellular respiration in the electron transport chain, they're going to be oxidized, and NADH is going to make 3 ATP, and each molecule of FADH₂ is going to make 2 ATP, and I'll show you why that is shortly. So once we're done with the Kreb cycle, we move to the cristae of the mitochondria. And so those chemical compounds are going to be sitting right here on the edge. And I want you to realize that on the cristae, we have an edge on each side of that inner membrane. And then we have a space called the intermembrane space as well. So each NADH and FADH₂ is going to donate 2 electrons. So let's look at that. If we start with FADH₂, it's going to lose 2 hydrogens, and each hydrogen is going to have a positive charge. And so to end up with the FAD, it then needs to lose 2 electrons. So it's going to donate 2 electrons. NADH is also going to lose 2 hydrogens, which would give it a negative charge because 1 hydrogen is going to go with 1 electron, but the other electron actually makes the final product have a positive charge, the NAD+. Okay, and so now we're going to have all these hydrogens sitting in this purple intermembrane space, creating a huge concentration gradient between the space and the matrix. And what's going to happen is the electrons that were released, they are these purple things, and they're going along these protein carriers, and they're going to be traveling down, down, down until they meet up with an oxygen molecule. And that's going to be the final electron acceptor. And so the oxygen is also going to pick up hydrogen molecules to form water. So over here on the right, you can see how that is. Here, the electrons being passed down. We're going to have 2 positively charged hydrogen molecules, and we're going to bring in 2 electrons to neutralize them. And we're going to bring in half of an oxygen to form H2O. And it's this kinetic energy of the hydrogen being very high concentrations there and very low in the matrix, and we're going to bring some into the matrix, that's powering the synthesis of making some ATP from the ADP with an ATP synthesis. As the hydrogen comes back across, here's our hydrogen coming back across, that is what's going to cause the conversion of ADP to ATP. And so this final little step is called oxidative phosphorylation. So remember, oxidative means it's going to involve oxygen, and phosphorylation means we're going to move a phosphate bond. So here is what's happening. We are bringing hydrogen across, and we are taking ATP and adding a phosphate to it. So we're phosphorylating it to make ATP. That's the phosphorylation part. Okay, and that is going to happen on the inner mitochondrial membrane. In the intermembrane space, we're going to have those electrons with the oxygen and the hydrogens forming water. Okay, so it's oxidative phosphorylation. And remember, Oxidation means we lose the hydrogen. You see over here, the hydrogens. All right, so producing ATP in this electron transport chain. So let's go back to these NADHs and the FADH₂. There are 4 little steps right here among that inner membrane. NADH enters at the first step. And so it passes through this hydrogen pump, this 3rd hydrogen pump, and this 4th hydrogen pump. And each time it passes through a hydrogen pump and it releases a hydrogen, it's going to generate an ATP. So NADH can make 3 ATPs. FADH only goes through 2 hydrogen pumps, number 3 and number 4. So that's why it can only produce 2 ATPs. So how much energy do we end up and where does it come from? Well, textbooks disagree on this a little bit. They go 34 to 38. Personally, I just say, let's stick with the 36 because that way the numbers are solid and you can understand it. But remember, we need to transport molecules from the cytosol into the mitochondria, and that's going to take some energy. All right, so let's go with a total net ATP yield of 36. And so here's the basics. From glucose to pyruvate, we're going to have a net of 2 because we use 2 and we make 4, so we have net of 2. And the Kreb cycle, we're going to make 2 ATP directly because we had to do it twice. And then we're going to make a bunch of carriers and an electron transport chain. If every NADH2 makes 2 ATPs and then the other one makes 3, then we will end up with 32 ATPs. So stop it for a second. FADH2, sorry, and NADHs. Stop this for a second, talk yourself through this equation. You need to write it down because me talking, I'm mixing up my electron carriers. And before you move on. Oh, and also don't forget to talk about what happens in oxidative phosphorylation. Don't just say, and oxidative phosphorylation. Go through that process so you understand what's happening. All right, so remember at the beginning, I said this is how all of our nutrients give us energy, and I only talked about glucose. Those simple sugars, glucose, galactose, they're going to do the same things, okay? Some other sugars are going to require some energy, or they may enter the glycolysis chain later on, so they may not give exactly the same amount. But let's leave carbohydrates aside because with glucose, we've got carbs covered. So let's talk about what happens with fats and proteins. With fats, remember, really should say lipids, but remember our lipids are glycerol and fatty acids. So the fatty acids, remember they're chains, and so they have to get broken down into 2 carbon chains. And what happens is those 2 carbon chains don't have to go through glycolysis at all. They can enter right there and use the enzyme CoA and form acetyl-CoA. And they only can do aerobic oxidation if they do that because they're not entering in the cytoplasm. And they do have a byproduct of keto acids, which we will talk about much, 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 much later in the course. In contrast, proteins have basically 3 different areas because it kind of depends on the amino acids. Well, one thing they all have in common is they all have to lose their amino groups. And basically, this becomes a waste product. It gets converted when it's in the blood and it gets to your kidneys. It's converted to something called urea, which your kidneys will excrete. We'll learn about that when we learn about your renal physiology. Now, the amino acids, remember, because some of them were polar and some weren't polar, and some were positive charge and some were negative charge, and some had long side chains and some had practically no side chains. So depending on the amino acid, some of them can enter in glycolysis and you will end up with pyruvate. Some can enter in the intermediate stage and some can enter in the citric acid cycle. So you cannot say amino acids always generate this amount because it depends on the amino acid. But I wanted you to realize that all the nutrients you consume can make ATP, the energy that your cells need to do their work. So let's go back and talk about fermentation. Why do we have it? We have it if there's no oxygen. So fermentation, the only thing we're going to do is be able to energy harvest from sugar without the presence of oxygen. And basically, that is glycolysis. So there's two different products we can make. At the end of glycolysis with a pyruvate, if we give off carbon dioxide gas, then we make ethanol. Okay, so think of carbonated beer, carbonated champagnes, or we can let the carbonation go out in the world and end up with our regular wine and those types of things. Now, you know our bodies aren't making alcohol, so that's not what's happening in our bodies if we don't have oxygen. What's happening in our bodies is the 2-pyruvate are converted to lactate, also known as lactic acid. So with fermentation, we only have a net of 2 ATP because that's all you get at the end of glycolysis. So think for a second, do you want from your nutrient, from your glucose molecule, do you want to make 2 ATP or do you want to make 36? You want to make 36 because that muscle contracting, every single little myosin cross bridge thing requires an ATP. So lactic acid fermentation of muscle cell, that's going to give you muscle cramps. Not a good thing to have. So what do you need to do? You need to breathe. A lot of people don't realize that when they're exercising, especially like if you're in the gym lifting weights, you're holding your breath. You're setting yourself up for lactic acid fermentation. So let's not do that. And then just because this is a biology class, let's get out of the human body and realize that pyruvate with some bacteria forms Swiss sauce, with a different bacteria forms all your cheeses and yogurts. And with yeast, you can form lovely breads, all kinds of things. So fermentation's not a bad thing, except for in our bodies. That's where it's not good to have. All right, so take a moment, go over the difference between cellular respiration versus fermentation, stop it. And now here is an overall view slide that kind of puts it all together, super basic. I typed out some core ideas for you to look at, as well as some review questions in case you like trying to answer questions. And with that, I am done with this very lengthy process, a multi-step process of cellular respiration. I want to thank you for all your hard work and putting in the energy that's required to be successful in this class. And I will see you in the next video. Bye-bye.